Amino acids are chemicals that have a very common sort of root structure, and that is an amino group at one end, a carboxyl group at another end, hence the term amino acids, bonded around this central carbon. This group up here in the red, that's going to change, and it's going to give the amino acid its identity. And of all the things that proteins do, they're built up of these amino acids. So the monomers are amino acids, the polymers are proteins, and something really doesn't become a functional protein until it has a three-dimensional shape. Now, as we said last slide, the amino acids are going to differ in their R group. Those R groups are going to have different characteristics, and that's going to give the protein its ability to interact with other amino acids and its environment. And here we see the different amino acids. We can group them. Many of them are nonpolar, and we notice that the amino acids, all we're changing again is the R group. Okay, represented differently. This is that central carbon before. There's the amino group. There's the carboxyl group. And this is what we put in where that R was. And it's going to give it its different name. We give amino acids a long name like tryptophan, or a short three-letter code, or a single-letter code. Now the single-letter code often comes with a lot of databases in genetics, because a lot of the proteins that are going to be made are going to be hundreds of amino acids long, and it gets a little tedious writing out the individual names, or even the three letters for that matter. So the one-letter system works. So we've got polar and nonpolar amino acids, so hydrophobic, hydrophilic amino acids, some that are bases, so they'll tend to become positively charged, and others that are negative, and they'll be acids in water-based solutions. Okay, so amino acids, because they have this central bonded carbon with its tetrahedral arrangement, and all four groups are different that are on that carbon: hydrogen, the carboxyl, the amino, and then the R group. That means we're going to have handedness here. We're going to have a left-handed amino acid, if you will, and a right-handed amino acid. And remember, we talked about isomers a little bit in class the one day. The example we used was thalidomide. This is maybe an easier example. It's not as extreme, but they're optical isomers. Enantiomers is another name, or chirality is the property that these things show. The left-handed amino acids are the 20 that you're going to see building up the proteins in all living things. So if something's made from a natural source, it's always made with the L amino acids. This is what living things want. They want their amino acids, the proteins, in this left-handed form. This other form, typically we don't see that in living systems, but we'll see it if something's artificially made. And again, amino acid is an amino acid, whether it's got, as long as it's got this same general structure, and you can put anything here, but it's 20 amino acids making up the proteins in all living things, just those 20. Now the bonds that join amino acids together are going to be called peptide bonds, and a single amino acid is an amino acid. If we join two of them together, we've got a dipeptide. 3 would be a tripeptide. If we do a whole bunch, it would be a polypeptide. But what links these things all together is going to be the peptide bond. And again, it's going to be condensation, or my preferred term, dehydration synthesis. And we're going to get water out. And again, we don't have two hydroxyl groups here, but we do have one on one amino acid. And we've got the hydrogen on the other. Now, some of you may be asking, well, why don't we use two hydroxyl groups like we did with the carbohydrates and with the lipids? If we did that, we'd be left with two amino groups at each end, and we'd have no way to continue this chain. We'd have no way to make it a polymer, so that makes it a little bit different. So what we're going to do here is we're going to take those two functional groups, and we're going to get our water out there. And when we do that, we're going to end up joining this carbon right to this nitrogen group, and we'll have a dipeptide. Now this means when we put proteins together and build complex proteins for amino acid, we produce water, but this reaction is reversible too. Now the enzymes are different, but so if you want to break apart protein, you have to ingest a whole lot of water, and that's the same thing that goes with carbohydrates or lipids. If you want to break complex molecules down, it takes water going in to do that. And this is why if you read the backs of energy bars and these sorts of things, they have instructions that say eat this and consume with plenty of water, and the water is used to break this bond. So not only does the water come out, it'll go in, and if the water goes in, we call it hydrolysis. That's something we're going to look at after we do macromolecules very, very briefly. So here's what these two amino acids are going to look like once we're done. There's our peptide bond here between this carbon and this nitrogen. This peptide bond is rotatable, so these two molecules can rotate, but typically the oxygen and the hydrogen tend to be shown as pointing away from each other. This also means this dipeptide has what we would term ends. It's got what we would call an A terminus, where the amino group is, and a C terminus, where the carboxyl group is, and that means this thing is going to have direction, which means now once we have direction and we've got differences, we've got something that's a little bit like a language that will result in the structure we see in proteins.